I'm pleased to be able to introduce Mike Newbrey, who most of you know since he's been here for about 4 years. Mike received his bachelor's degree from the University of Wisconsin, and then continued on there studying in a degree in fisheries. So he's got both the paleontology and the biology background. And for his master's degree, what he did was look at submerged trees as fish habitat, and look at it from a biological point of view. He moved to North Dakota State University to do his doctorate, and for his doctorate, what he looked at was growth patterns and the relationship of growth patterns in various fish to climate, and with the ultimate goal of looking at the relationship of this to climate change. And this leads into the talk that he's going to give today, which is on this topic in sharks. So take it away, Mike.
Thanks, Don. So as you heard, my talk today is about sharks, entitled The Effects of Climate Change on the Growth Biology and Geographic Distribution of Cretaceous Sharks. This is a topic that I have an interest in, not only from the standpoint of sharks, but also from the standpoint of studying bony fish. Now, shark teeth are fairly common in the fossil record, and they're common for a variety of reasons, but they produce information about diversity, origin, extinction, evolution, and feeding ecology. They can't tell us a lot about the rest of the organism because it hasn't, in most cases, been preserved. Shark teeth represent hard parts of shark skeletons that get preserved, and they're also produced on a regular basis as sharks shed their teeth and grow new ones, and they're constantly replacing their teeth because they wear out. Here you see a large megatooth shark tooth right here from the Cenozoic, and lots of teeth scattered around that. Now, teeth, as I mentioned, can provide a variety of information, but in rare instances you find sharks that are preserved as skeletons. And it's a bit difficult to see here. We're having projector problems. So the head is a little dark, but the anterior of the head is down here, and the rest of the fish would extend up this way. And you see the head here. And then there's a long vertebral column. This is a 5-meter-long shark called Cretaceous. And all these individual vertebrae are stacked together, and they're sort of knocked over like a bunch of dominoes. Now, these things, we can take a look at vertebrae, and they can provide a lot more information about shark biology than just teeth alone. So, for example, we can take a look at a single centrum, section it, age it. We can figure out how long this individual lived, how fast it grew, how big it was when it was born. We know that this is a 5-meter shark, and from age and growth studies, and this is hot off my desk, you guys, we can back calculate the size to 105 centimeters in total length at birth. That's pretty cool. We can do these sorts of things with centra and a whole lot more. So when we get complete skeletons, or even isolated centra with teeth, you know, they're rare, but they provide a lot of information, so they're worthy of study. My talk is based on this outline today. I'm going to introduce fish. I'll give you a brief background on shark diversity, and I'll talk about the 9 orders of sharks. We'll discuss why sharks are important to study. We'll introduce climate change and how it impacts the biology of bony fish. We'll introduce some late Cretaceous sharks and examine the responses of these sharks to climate change. Now, Joe Nelson in 2006 in Fishes of the World published this estimate of 28,400 species of described fish. They come from all over the world in a variety of sizes. They feed on just about everything you could possibly imagine. They range in size from being 8 millimeters long, like this goby from the Indian Ocean called Trimeton nanus. And the largest fish we have on the planet is a species called the whale shark, Rhynchodon typus. And here's a diver here for scale. They range in elevation up to 5,200 meters in Tibet. This is Yananilus altus, and we have the river loach pictured here instead, which is a closely related species. And they range down to depths of 7,000 meters below the surface of the ocean. This is an undescribed snailfish. This image was released last year. It still hasn't been described. They're found at temperatures of 42.5 degrees C. This is pretty hot for a fish. In hot soda lakes in Africa, for example, Lake Magadi, Kenya. This is a soda tilapia. It's a kind of cichlid. They're also found at temperatures below freezing, believe it or not, under the Antarctic ice sheet. This is the emerald rock cod, and they have antifreeze in their blood. This is a hot topic for study in physiological studies that are ongoing at the moment. And then we have the Elasmobranchs. This refers to a subclass of fish called the Elasmobranchii. The term elasmobranch is actually very descriptive for these fish. We have sharks and rays, and they're made up of cartilaginous skeletons. The term can be split into two Greek words. Elasmos, I'm sorry that's hard to see. Elasmos being the Greek term for thin, flexible, beaten metal. And branchios, meaning gill. And I like to think of these fish, or this term, as stiff but flexible gills, which describes the cartilaginous nature of the skeletons. Rays consist of 4 orders, 34 families, 106 genera, and 534 species. It sounds impressive, but it only consists of 1.8% of all living fish today. This is not a particularly diverse group of fishes, but there's some very interesting ecologies represented within it. This is the shovel-nosed guitarfish, Rhinobatos productus. And here is a fossil from Alberta. Now, I'm going to say something shocking. This is, in my opinion, the coolest fossil that Alberta has produced. Now, I don't want emails from the dinosaur people. I don't need that in my life. However, this is cool because this fish has an articulated dentition and a skeleton associated with it. Up until 1998, when this was found, we only had isolated teeth to work with. We didn't have any complete tooth sets, let alone complete individuals. And Don and Andy's work and Andy's work have, now Andy Newman and Don Brinkman, for those of you who are not familiar with them, have classified this as guitarfish, based on the continuous nature of the pectoral fin with the head. So the head is here. There's an enlarged pectoral fin, part of the disc. There would be another pectoral fin over here. And this is the view of the underside. Here's the mouth. This would be the right lower jaw, called the right Meckel's cartilage. This would be the right plateauquadrate. Here's extant Rhinobatos. You can see the top view, but not the mouth underneath. We see this dentition here, and we have a pretty good idea of how the dentition is put together. This is actually a picture from the top side of this fish. These are replacement teeth, and you can see the dentition here on the jaws, and you'd be able to take a close look at these. This fish is on display in the 25th. I highly suggest taking a look at it. When you do, you'll see these teeth in the jaws. They're all worn smooth. They grind together and wear off the occlusal surfaces that you see here, so you won't see hardly any of the enameloid folds or the transverse ridge that are characteristic for these teeth. But like other sharks, they're constantly replacing their teeth, so they have this conveyor belt style tooth replacement. And these are teeth that are not functional, and they're still waiting to move into a functional position in the mouth. Back here we have the pectoral fin, or rather, I'm sorry, the pelvic fin, and the vertebral column. We have branchial arches here. And on Rhinobatos and many other sharks, we have claspers on the males. These are elongate calcified structures. You can't see that here. This area has not been prepared yet. So very soon, we'll be able to prepare this out and sex this individual and determine whether or not it's a male or a female based on the presence of a clasper. The work that I've done with this so far would suggest this is a 5-year-old adult with an estimated length of around 110 centimeters. This age and size is quite comparable to Rhinobatos today. So there's my plug for the coolest fossil in Alberta. I highly suggest going and checking it out. And from here on out, we'll talk about sharks. Now, there are 9 orders of sharks, 17 families, 72 genera, and 403 species. It only comprises 1.4% of all living fish today. And I'm going to introduce you to the 9 orders. The first one being the heterodontiform type sharks. These are the bullheads. And the term here, hetero, meaning different, and from the Greek term heteros, and odonus, odus, meaning teeth in Greek. So they have teeth in their mouth that are, have different morphologies. And they have these interesting screw-shaped egg cases. They attain about 1 meter in length and inhabit the tropics. You have the carpet sharks, the erectolobiforms. This group contains the largest shark that we have on record, the whale shark. They can exceed 12 meters in length and can get up to about 20 tons in weight. They have a tropical distribution. They're filter feeders. We have the white shark representing this order, the lamniformes. These are the mackerel sharks. White sharks often swim in schools of fish, especially the smaller ones. And this particular school here are pilchards. They're a sardine. And white sharks will generally feed near the surface of the bottom. They'll be found down to over 1,200 meters in depth, or right at the surface, an amazing range for a fish, I think. And medium-sized individuals will often eat fish. They'll eat invertebrates, believe it or not. Off the coast of Washington State, there was one white shark recovered with 150 crabs in its stomach, along with some salmon. Larger individuals will feed on pinnipeds. So you've all seen footage of sharks flipping seals up into the air and then grabbing them and toying with them. That's something that sharks do not only with seals but with birds. And they've been known to feed on large cetaceans, so they will feed on dead dolphins or dead whales. We don't actually know if they feed on live ones. That hasn't been well studied at all. Another iconic shark, in addition to the last one, is the tiger shark. This is a member of the order Carcharodontiformes and the ground sharks. It's the most diverse group with 224 species. They have a tropical distribution. Tiger sharks will attain lengths of around 5 meters. However, there is one documented report of a tiger shark being 7 meters long. It was a large female. We have the hexanchiforms. This is a less derived member of the living sharks today. This one was photographed at 1,800 feet in depth. We have the bramble sharks, the echinorynoforms. The name echainos in Greek means spiny, and rhinos in Greek means nose. So this actually refers to the nature of these fish being having a spiny nose. You see these little freckles all over them. These are actually little patches of spiny denticles. There's the squaliforms, which are the dogfish sharks. Many of you have taken comparative anatomy classes if you work in this museum on fossils. And these are commonly used to study anatomy. We have the squatiniforms, the angel sharks, not to be confused with the rays in that they have a terminal mouth, even though that they have a fairly flat body. The rays, as you remember, have the mouth underneath. And it's difficult to see from my angle, but here's an angel shark right here in the middle. I don't know how well you can see that in the back, but these things will bury themselves partially in the substrate to avoid predation and to ambush prey as they come by. We have the Pristophoriforms. These are the saw sharks, not to be confused with the saw fish. These have a long rostrum with small conical teeth on them, a very pronounced pectoral fin. Sawfish, which are a type of batoid, have also a long rostrum, but very wide, flat, double-bladed teeth running along the rostrum with a pectoral fin that's more closely articulated with the head. Now, sharks are important to study in our environment, for our society, and are important ecologically, economically, with regard to us eating them as food for a recreational fishery and for ecotourism. They represent top predators in our ecosystems and are found along the food chain in a variety of places. This is the tiger shark here. They can feed on fish. Here you can see a large bite mark out of one, or occasionally on surfboards. According to Lowry et al., the shark that made this bite mark on the surfboard would have been about 4.5 meters in length. That was a study on bite circumference. So these things do act as predators in our ecosystems. They greatly influence the food chain through controlling the abundance of their prey. This is called top-down control in the food pyramid. They eat carrion, which serves as a vital role in cycling nutrients in our oceans. And as you know, we dump lots of garbage and waste into our oceans. We need healthy oceans. So we need these sharks there serving that role. There's my punch for conservatism there. This is a shark that's being butchered for food. It's a whale shark. Sad thing is, we don't even know how long these things live. We have no idea about their growth biology, yet we're out there harvesting them. We harvest sharks for their fins to make shark fin soup. Annually, 100 million sharks are killed for their fins. 1 pound of dried shark fin meat can retail for $300 or more. Oftentimes, fins are cut and the sharks are just tossed overboard to suffocate at the bottom. We fish for sharks for fun. This is the white shark here and a tiger shark. And fishing is actually quite popular, especially off the coast of Florida. In 1992, the shark fishery in the Gulf of Mexico alone, near Florida, was worth $66,220,000. And after 17 years of inflation, and this is hard to see, it equates to about $100 million in 2009. And Florida has the highest active commercial shark, or rather the biggest active commercial shark fishery, accounting for over 4.8 million pounds of sharks harvested each year. Ecotourism plays an important role in the conservation of sharks and putting money into the study of sharks. We like to dive with them. We like to stand or dive in cages and watch them. And certainly this puts a lot of money into the field. However, I haven't been able to come up with a value for this yet. Now we're pushing many of our sharks to the brink of extinction through harvesting them, through sport fishing, and in other ways. And despite many years of study of sharks, especially this iconic creature here, the white shark, we know relatively little about sharks. And that's really interesting and spooky, because we've been studying bony fish for a long time. We know quite a bit about them, especially bony fish that we like to eat or that we have recreational fisheries for, northern pike or walleye or yellow perch, for example, here in Alberta. Now the white shark, we have no idea how long these things live. We know they live at least up to 22 years old. Estimates would suggest 50 years old, but we just don't know. We're out there pulling out these big fish out of the ocean. We actually don't know how long some of the biggest individuals live. We don't know how big they get. We have documentation up to 5.6 meters. We have estimates, questionable estimates, up to 7 meters. We have undocumented reports up to 9 meters, ranging up to 9 meters. We actually don't know how long they live. We don't know about their litter size. We know they can produce at least 2 pups. We found individuals with 14 undeveloped pups inside of them. So we know they can produce perhaps up to 14 pups. Now what we know from bony fish and many other marine fishes is that larger individuals can produce more offspring. So if you have large individuals that have been yanked out of the breeding population due to fishing or harvest or whatever, you know, what effect does that have on replacement for these in their various groups? And how would climate change affect that? And that's something I'll look at today. So, climate change. I'm just going to introduce this in a quick 5 slides. So this is your crash course if you haven't been exposed to this. We'll talk about climate change from the perspectives of the geologic record and our current observations. So a quick overview. This is a time slice from 97 million years ago to about 71 million years ago. And this is a paleotemperature curve. It would represent sea surface temperatures in the UK. These temperatures have been derived from oxygen isotope studies from deep benthic foraminifera. And what we see here is a long global warming trend and a long global cooling trend, with a peak in temperature here at the Cenomanian-Tronian boundary. Based on some of Don's work, we have animals like turtles and champsosaurs living at these peaks here in the Arctic. There's no freezing temperatures there at the time. So clearly, this is a very warm time in the fossil record. Now, on this curve, there's a lot of temperature fluctuation. And you see a lot of little global warming, global cooling events. These are actually profound temperature change, yet they fall on an overall pattern of global warming or global cooling. And we have fossils scattered throughout this time period. So we can start to pose questions about, well, what happens with shark biology with climate change? So another paleotemperature curve. So here we have relatively warm temperatures at the Cretaceous tertiary boundary when the dinosaurs went extinct. To put it into context, average temperature in North Dakota at that particular time was 8 degrees C. Here's the early Eocene thermal maximum. We have warm organisms living on Ellesmere Island in Canada. We have tortoises and alligators, so non-freezing temperatures or about freezing. We have a long cooling trend here. We have ice forming at the Eocene and Oligocene boundary in Antarctica. We have the Miocene thermal optimum and another long cooling trend down to 1.8 million years. And then since then, temperature has been fluctuating back and forth with glacial patterns. Shackleton et al. Estimated maybe perhaps 50 glaciation cycles in North America. A little bit more on climate change. And from the geologic record, we can look at the concentration of greenhouse gases in little bubbles of ice. And atmospheric carbon dioxide levels are strongly correlated with Antarctic temperature over the last 800,000 years from glacial ice. And at times when carbon dioxide levels are high, it's very warm in Antarctica. Now, current CO2 levels exceed those over the last 15 million years. So clearly, there's a problem there with carbon dioxide. Methane levels are 1,700 parts per billion by volume, and over the last 800,000 years fluctuated between 3 and 800 parts per billion. So they're very high. Now, these greenhouse gases, they can influence our global temperature. So, for example, the sun emits solar radiation, long-wave infrared radiation hits the Earth. About 40% of it is re-reflected back out into space. Now, due to anthropogenic processes, we've dumped greenhouse gases into the atmosphere, and these molecules in our atmosphere are intercepting the reflected radiation, dispersing it and bouncing it back to the Earth where it's absorbed and re-radiated as heat. So we're heating up our climate in this manner by the production of these things through deforestation and burning of fossil fuels. Based on what we see today, we can say that Arctic temperatures have risen about 1 degree centigrade in the past 2 decades. And Arctic sea ice is declining at 11% a decade, according to the National Snow and Ice Center. So some climate change experts are forecasting that the Arctic summers will become ice-free very soon, and that's pretty scary. So what do we know about the effects of climate change on sharks? As I mentioned earlier, absolutely nothing. And what does that mean? It means we could be in trouble when it comes to dealing with our fisheries. For example, if we're harvesting many of these fishes, we don't even know at the rates we're harvesting them. We don't understand their biology. And if we have climate change, could this impact the rate at which many of these things start to disappear? And as you're probably all well aware, bringing species back from the brink of extinction is extremely costly. We do not want to push them that far. The ecological ramifications of losing a species are nightmarish in my mind. So let's look to the bony fish. What do we know about the effects of climate change on freshwater bony fish? This is a topic that I've looked into, as Don mentioned earlier, and I've done work. And I just want to address a couple of questions. Does fish distribution vary with climate change? We'll try to make some inferences about what we could predict with sharks. And does age and growth vary with climate change? And the quick answer is yes, they do. So fish are ectotherms. They respond to the water medium that they're in that they have the same body temperature as the surrounding water. Now, ectotherms respond to Q10 relationships, and I'll explain that. For every 10 degrees centigrade increase in temperature you have metabolic rates increasing 20 to 30 times, and reaction rates of mussels increasing 100 to 200%. That's huge. So ectotherms can respond to temperature in a very major way. And then how they do that is variable amongst the taxa that you look at. And I'll show you that in a moment. Now here's an example of a tiger muskie. This is of the genus Esox. Esox feeding rates have also been known to increase with temperature, and this can be modeled. And that study has looked into that. These things can be applied to sharks as well, even though we haven't studied them. Sharks are ectotherms. Some sharks show evidence of endothermy. We won't discuss that here. So what would our predictions be with bony fish given climate change and across the range today of these organisms? So we would expect that during globally cool events, fish distribution would be shifted further south into warmer latitudes. And during globally warm events, that they'd be shifting further north, tracking with climate change, because fish have thermal optimal ranges. They have a thermal optimal range for growth, for feeding, for spawning, and a whole suite of things. Yes, this is a spooky graph, but I can break it down into something fairly simple. We have fossil localities colored in yellow. These are all the symbols you pictured here, and they correspond to this axis here in latitude. So here are localities in Alberta around 50 degrees latitude at 60 million years ago. Here's some fossil localities in Wyoming at 50 million years ago, around 40 degrees latitude. Here's that paleotemperature curve you saw earlier, the early Eocene thermal maximum, ice forming in Antarctica. It's red on this axis over here. I just moved it to the other side at these particular ages. Now what I want to do is correlate these latitudes against temperature and test the hypothesis whether or not Esox in the fossil record is responding to climate change. And in fact they are. And there's two hypotheses tested here. One for Esox. This is during the last 65 million years. No matter what age you're considering for a locality, during warmer times, Esox are found north in cooler latitudes, and vice versa during cooler times are found in warmer southerly latitudes. Same thing can be said for North American Hyodontidae during the last 65 million years. So what we might predict is that sharks would be shifting their distribution into higher latitudes during warmer times. For growth, I know this is an equation. I know many of you hate math. But let's just break it down into a couple of simple components. Total length at a given age. If I was a fish, we would say that Mike Newbrey has, we'll just call it total length or a height, total length of 6 feet at age 21. And now length infinity is the maximum possible size in a population. So say my relatives breed with you guys and this room is a lake. So who's the biggest individual in here? I'm going to guess this is Andy Newman. He's got to be over 6 feet. So we'll use Andy Newman as a model for a maximum possible size. Growth rate, this is just the average growth rate, how fast we grow all averaged together. And inherent within T, this is time in years, is lifespan. So who's the oldest fella in here? Dennis Brayman, perhaps? We'll call this the Dennis Brayman factor. So this is T. If we know how old Dennis is, how big Andy is, how fast we all grow, we can predict my size at a given age. That's all this thing does. The other factor here that we haven't considered, that von Bertalanffy didn't consider in 1938 when he developed this equation, is temperature. Now, I can correlate any one of these variables with temperature, and we can look at the effects of temperature change on fish. Not only can we do it with living fish today, but we can do it in the fossil record. So, for example, this is age in years lived. This is total length of northern pike, and here's 2 northern pike. Each line is a population of fish from a different spot in North America. Here is a Canadian northern pike from the Northwest Territories. It's about 35 centimeters in length at age 4. Here is a pike from Ohio. This is about 75 centimeters in total length, and the mean annual temperature there, or the average temperature, is 10 degrees C. It's a lot bigger than this one from the Northwest Territories, where temperature is -7 degrees C. Now we can plot sizes at age 4 for all these populations against temperature, and we get a relationship that looks like this. So these are all lakes and rivers that have northern pike along this line. Here's the Northwest Territories here, here's Ohio here, here's Oklahoma up here, for example. And down, or rather up north, where it's quite cold, at age 4, northern pike are quite small. But way down south in Ohio, in Oklahoma, they're much larger at age 4, where the temperature is roughly equivalent to 15 degrees C. The same thing can be said for muskie in Ontario, where they're much smaller versus those in Tennessee, and the chain pickerel. Now, I could have plotted maximum size here. I could plot growth rate. I could plot Dennis's lifespan, so to speak, and we can model these things against temperature and find lots of interesting, significant relationships to make predictions about climate change. This is from extant data, so we can make predictions from it. We can say this is how they could respond to climate change, but what about how they do respond to climate change? This graph shows both sets of data. Now, here's longevity. This is age in years lived, the Dennis factor. We have northern pike. Up north in the Northwest Territories, they live 20 to 25 years. Down south, they only live 5, 6, 7 years. That's their whole lifespan. For muskie in Ontario, they live a long time, 25 to 30 years. Down in Tennessee, they only live 6, 7, 8, 9 years. Much shorter lifespan. In the fossil record, we see the same trend. It's just offset. During globally warm times, they have very short lifespans. This is data over the last 65 million years. And it's a significant relationship. Notice that this whole data range is offset from the rest of this. There's a significant difference there. This indicates adaptation to climate change. These are adapted for warmer thermal environments. These are adapted for the cool thermal environments that we have today. So it's interesting to think about this in terms of these short-lived fish down here don't have the same number of lifetime reproductive attempts as fish up here. How would that impact sharks if we saw a relationship like that? So just to sum up some of my work in text, the latitudinal distribution of esox and hyodontid localities is positively correlated with temperature, as we would expect for sharks. This still needs to be tested, though. And there's multiple relationships that exist between temperature and growth characteristics for both extant and extinct taxa in both families examined. In 17 out of 20 relationships examined, in 3 out of 5 relationships examined, and there are significant differences in growth characteristics between extant and extinct taxa, indicating adaptation, and there's 9 out of 11 relationships examined. 9 relationships showed that pattern. So fish are responding to climate change in the fossil record, and based on their biology today, we would expect them to do just the same. So what about sharks? How do they respond to climate change? Let's take a look at 4 groups and try to make some inferences about that. Lonchidion selaikos, Cardabiodon ricci, and Cardabiodon venator, various squally corax species, and Cretaceous mantelli. Now, Lonchidion has a fossil record that extends from 65 to 99 million years. Here you see Lonchidion teeth and fin spines. Lonchidion selaikos teeth occur in freshwater deposits from North America during the Upper Cretaceous. And we'll test the hypothesis that geographic distribution of Lonchidion tracks with climate change, as we did with northern pike and hyodontids. And here you see very typical Lonchidion teeth. They're T-shaped in occlusal view. They have a surface on the side of the lip, the labial face, that's smooth. They have a strong lingual protuberance. The roots here are missing. They have serrated fin spines. We'll look at Cardabiodon. It has a fossil record that extends from 91 to 95 million years, a very short time. Cardabiodon is from marine Cretaceous deposits in Canada, the USA, the UK, and Australia. And we'll test the hypothesis that geographic distribution of Cardabiodon tracks with climate change. And Cardabiodon has a lot of interesting characteristics, foramina that are scattered across a root, compressed root lobes, labially directed little cusplets, little heels next to those cusplets, a big cusp. And this is the holotype from Australia. And we estimate the bite, half the bite circumference to be 360 millimeters. This would suggest a shark that's about 5.6 5.6 meters, sorry, in total length. We look at Squalecorax species. This is a Squalecorax specimen that I examined in the Smithsonian. And all Squalecorax species have serrated teeth on the main cusp of their teeth. We looked at a variety of taxa. We used a parataxonomic system for identifying these things. All of Agassiz's old names are left in nomen dubium as far as Syverson et al. In 2007 is concerned. And then he picked on specifically Squalecorax falcatus. But Squalecorax extends in the fossil record from 65 to 105 million years about. Samples are from marine Cretaceous deposits in Canada and the US. We have centra extending in the fossil record, extending from ranging from 80 to 86 million years. We test the hypotheses that Squalecorax size at age 3 and longevity are correlated with climate change. And specifically, we'll use these groups here. These other groups here are particularly small and have very different biologies and morphologies to their teeth. We also look at Cretoxyrhina mantelli. It has a fossil record that extends from 83 to 99 million years. Samples are from marine Cretaceous deposits in the US. Centra range in age from 77 million years ago to 95 million years ago. And we test the hypothesis that Cretoxyrhina size at age 3 and longevity, as in with Squalecorax, are also correlated with climate change. And here's that first data set of the results. Here's paleolatitude of fossil localities on the primary y-axis. Here are fossil localities. Here's temperature, as we saw earlier, and as you can see, the fossil localities seem to sort of track with temperature, and that they're moving south into warmer latitudes. But to formally test this, we need to correlate these data points across these two axes, and we do that here. And what we see is a significant relationship. During globally warm times, Lonchidion is found in fossil localities further north, and during cooler times, they're found in localities further south. This is the first evidence that I presented to you that sharks are responding to climate change. What about sharks in the open ocean? Now I'll show you a study that was published last summer. Actually, it was data from that study. We never actually drew a conclusion regarding climate change. But the samples that we've included are from Australia, the UK, and North America, including Watino, Alberta. And this is their response to climate change. Both species are found at higher latitudes in the northern and southern hemispheres during globally warm times. These things are shifting their geographic distribution with climate change. And the paper was based around testing the hypothesis whether or not they had an anti-tropical distribution. But in this particular instance, the data also works to make this conclusion as well. And this is what we predicted from the bony fish. Now, what about age and growth? Here we have all these vertebrae. We can start to age these things and then look at the effects of climate change on age and growth biology. So this is a vertebral centrum from Squalecorax. We used a sagittal section that runs right through the middle. This is the birth ring. This is the size of this centrum when this shark was born. Of course, it was living inside of its mother and growing before that. Here's the center of that centrum in side view. And you can find the birth ring here, a textural change at the surface. Typically, there's an angle change here, which is almost more apparent in this image, but the centrum has been sort of compressed and crushed a bit, so it's harder to see that. And then here you have all these rings that we count like we would counting growth rings on a tree. And we can say that this had an age on this arm of the corpus calcareum up to 17 years. It's broken. Down here it's at 18 years. And we can take and measure from the center out to each ring and plot that on a graph. And that's what I've done here. So here we have age in years lived, centrum size, and each line represents the growth profile from a single centrum. All the white lines are Cretoxyrhina. The gray lines are Cardabiodon. The yellow lines are Squalecorax. And this purple line is Archaeolamna. This is actually a tooth from the image of the title slide. You saw a whole bunch of teeth there. So we see that these things can live up to over 30 years in age for Cretoxyrhina and for Squalicorax, up to 12 years old for Cardabiodon. We only have 2 samples there. Archaeolamna copengensis was not included in this study. We only have 1 sample. But we do have more data for Cretoxyrhina and for Squalicorax. Now, if we take a look at size at age 3 and compare this to global climate change, based on the information we saw from northern pike, we should expect a significant relationship. And what we get is no relationships at all. There is no response using this data set to climate change. Now, I can poke holes in this analysis. This is actually a very coarse thermal data set to use for testing this hypothesis. But the same coarse thermal data set produces very interesting results for longevity. And they're real results. We could refine this analysis and clean up these relationships if we want to, and we will. Here's Cretoxyrhina mantelli. In globally warm climates, they have lifespans half the length of those in globally cool climates. A very similar relationship for a variety of Squalicorax species combined. This is profound. If we have sharks living today that show the same sorts of patterns, they're going to have fewer reproductive attempts in their lifespan in a warmer climate. Now, if we're harvesting them like mad, that could really impact these populations. So to sum this up, the distribution of 2 taxa of Cretaceous sharks were positively correlated with temperature change. And there's no relationship between size at age 3 and temperature, but there is a significant relationship, a negative relationship between longevity and temperature. And in the light of global warming and overharvest, sharks could be pushed to the brink of extinction very quickly. This is important information, and I realized this the other day when I was putting together these slides that I really need to get a manuscript out on this, because it's almost a disservice to the rest of the scientific community not to be aware of these sorts of hypotheses, so they can start to design studies to test these relationships with modern fish. And my work will still continue with fossils, studying teeth and whole skeletons to learn more about living sharks today. So with that, I'll wrap this up by thanking the Royal Tyrrell Museum Cooperating Society, who's funded me graciously over the last several years, and for the 7 travel grants that they've given to me to visit various museum collections like the Smithsonian, Los Angeles County Museum, University of Kansas, and others. And the discovery grants to Mark Wilson and Allison Murray, which have also contributed to this project. And I would thank these people from those various collections, and I'd like to draw specific attention to Allison here at the museum. She has perfected the techniques in sectioning these centra. Until she did this, we could not do this study. Many of these centra were just too fragile to take a good close look at age and growth. So I would like to say thank you, Allison. And with that, I will take questions.